Hello, class. Today our notes are going to be over mitosis. To start things off today, to give you a general overview of what this video is going to contain, the general proceedings are going to be that we're going to look at the different parts of the cell cycle first, which was one of our vocabulary words, where basically the cell preps itself to divide into two new cells, which is something that Virchow came up with when he was studying cells, one of the scientists that we already mentioned so far this year. During the cell cycle, things occur in three different steps. The very first step is a step called interphase. And for each one of these, we're going to look at three different components or pieces that make up each stage. So in each one of these, you'll kind of pick up a theme that there are three main facts that go along with each one. Now, some of them kind of seem to repeat themselves because I tried to come up with three different things for each one. For the first stage, the first stage is known as interphase. And what interphase does is that one of the main things is that this is your normal everyday type of cell. So it's going to allow the cells to grow to their mature size, which means the full cell becomes itself. We start off as, little tiny baby cell, and then they just keep on growing and growing and growing until they reach their full size. Now, they won't get too big, because if they get too big, then they can't carry out their major functions. It would take too long to transfer proteins from one side of the cell to the other. So there are drawbacks for being big cells. So that's why we're not one gigantic single-celled organism, but we're made out of trillions and trillions of cells that go into making us up, because they got to stay tiny in order for them to do their functions. The second thing that interphase does is that it makes a copy of its DNA. And that's the DNA that would be found in the nucleus. It's also the chromatin, which we'll be talking about a little bit later, and we'll be talking about a lot. And what chromatin is just the DNA. It's another word that we use for DNA. A lot of you have heard of the word chromosome. That's condensed DNA. That's like in its travel form. Right now, the cell is making a copy of it because in order to make two cells, we need to make sure that each of those new cells needs to have the same amount of information in each one. And each new cell needs its own individual copy of the DNA. The third part of interphase is that the cell prepares to divide. So that means not only does it copy the DNA, but it also starts to copy all the other organelles. So one of the things that will happen is that it will make another mitochondria. Or another chloroplast if it's a plant cell. It'll make sure that it has enough cell membrane for when it splits, that it splits with enough cell membrane in each one. It basically allows so that all the organelles duplicate as well. The second step of the cell cycle is mitosis, which is one of the main things that we're going to look at today. Now, for mitosis, I'm going to give you three steps to make it kind of simplified, because we're going to look at a lot of phases that happen during mitosis in a little bit here. One of the main things that happens during mitosis is that the chromatin, which is kind of this noodly-like DNA structure that is found inside the nucleus, so again, it looks like the string, it's just a whole bunch of it. It's all loose inside of the nucleus. That is going to transform into what I call the packaged version, which is what's called a chromosome, which you might be more familiar with. It'd be like this here. It's a nice piece of carry-on luggage. And since the cell already made a copy of its DNA, usually they come in two little bars like this. Typically, you'll see this represented as an X. So sometimes you'll find it, in the past you may have looked at things as an X. Those would be our chromosomes in this case. So essentially the first step is that the chromatin, which is this noodly stuff, transforms itself into the chromosomes. The second part is that these chromosomes, which is typically in an X, they end up going to opposite sides of the cell, because each one of these chromosomes end up having the same DNA content inside of them. So one of them goes one way, one of them goes the other way. Now what happens is that these chromosomes go into the new nucleus, and two new nuclei develop, which if we look at this little poster here towards the bottom, D here, this would be kind of that final stage here. On one corner you would have some of the chromosome going into it. There you go. Chromosome in one of those, and then you'd have another chromosome on the other side. That would form around it a nucleus, which would end up giving us a new cell at the end. Now the very last step of our cell cycle is the fact that cytokinesis happens. Now the word cytokinesis, you may recognize the first part of that, cyto, because we've already talked about an organelle that has cyto in there as well, cytoplasm. Kinesis is kind of the activities. Hiya. That was my karate chop there. What cytokinesis means is the cytoplasm gets cut. And that's what ends up happening during cytokinesis, is the cytoplasm ends up splitting in half. Now with going back to this picture that we'll look at a little bit later, over here in D, this is where the beginning parts of cytokinesis will happen. As you can see here, this is one cell trying to become two. In the middle here, you can see that it kind of pinches in the middle, and that's what ends up happening to our animal cells. They end up getting pinched closer and closer and closer in the middle until eventually bloop, it pinches enough to where we get two separate cells, which is over here at E. So the end result is that we get two new cells at the end. And these two new cells are typically called daughter cells because they came from a mama cell. And they also have identical genes inside of them and an identical DNA inside of them because they came from the same cell to begin with. So in order to have the since the DNA got replicated or copied in the first place during interphase, each new cell has its own copy of the DNA that's the same thing as the first one. So they're identical, which is why they're called daughter cells. For the second half of this video, we're going to look at the actual phases of mitosis. Now for the phases of mitosis, some of this stuff might sound familiar to some of the things that we just went over in mitosis itself. And that's because this is the breakdown of what mitosis is, because it happens in a series of phases. Now there's 4 phases to mitosis. We'll cover each one of them here. One of the phases isn't technically a part of mitosis. It's called interphase. We've already went over that. But most people include it in mitosis anyways because it just fits together with it. But the first actual phase of mitosis is a phase called prophase. The prophase, and for each one of these we'll do the same thing that we did up above, where we're going to come up with 3 facts that happen in each one of these phases. Some of them may be more important than others in as terms of what happens. Now the first thing in prophase is that we take our tangled mess of DNA, our chromatin, and we're going to condense it into its travel form of a chromosome, because it makes sure that and when this condenses, what it makes sure that happens is that none of the DNA gets damaged when they move, which is kind of like when you pack things. When you move houses, you pack everything into boxes and into suitcases and luggages, so that when you take them from place to place, none of your stuff gets lost. Same thing happens with the cells. It takes all of its important DNA information that's in its untangled, unpackaged mess here, and it's going to put it into a nice little carrying case for it, to where it shrinks it down and packages it so that it's very small, so that it can fit and gets moved easily. Whereas some of this stuff, some of it can get lost because as it stretches, maybe it'll cut off this part. And then all of a sudden you're missing that much of your DNA, which could cause your cells to malfunction. Or you could become sick from that. It's one of the things of how cancer develops, is this mitosis process going wrong. So you don't want it to happen. So the first thing is that chromatin condenses into chromosomes. The second part is that our centrioles start to move to opposite ends of the cell. As we mentioned as part of the organelle notes, centrioles, their main use is in cell division. It helps with the division. And they're going to become increasingly important here in the next upcoming steps. The last thing that happens is that the nucleus of the cell breaks down. It breaks apart because in order to get to the gooey, nougaty center that is the DNA, to get them to be packaged and moved to opposite sides of the cell, we have to have access to that DNA. And we only have access to that DNA if we break apart the nucleus. So we kind of break it apart to get inside so that we can get all the parts necessary that we need. Now, how that would look. This is going to go in picture A here. And we're going to just kind of draw along with it here. So we got our cells. So you should draw your cells here. So A, we're still just dealing with one cell. What we end up having is in blue here. The blue represents our nucleus breaking down. So a dotted line, it starts to break down. On the inside, which I'm going to show as orange, we're going to have chromosomes on the inside. So those orange things are chromosomes that are still inside the nucleus. We can finally see them because the nucleus is starting to break apart. And lastly, I'm going to use with purple the centrioles. And there's typically two centrioles, and they're going to be moving towards the opposite sides of the cell. So up top here you have your two centrioles with your two dots there, and the arrows that are moving to opposite sides of the cell. Our next step in mitosis is called metaphase. Now metaphase, one of the main things that you need to know about metaphase is that everything is focused in the middle of the cell. M for metaphase. Metaphase, middle. Meta typically means middle anyways, so we end up having a similarity there. It's the middle phase. It's in the middle of the whole cell process, and a lot of things are going to revolve around it being in the center and in the middle of the cell. The first step with metaphase is that the chromosomes all end up lining up in the middle of the cell. So we end up having all the chromosomes directly in the middle. What also happens is that the centrioles end up attaching themselves to each one of the chromosomes. Now I'll start drawing this for you here. By this point, all of our nucleus has gone away. So what we end up having is in the very first part here, this is B, we end up having our orange chromosomes all line up in the middle there. The second part is that the centrioles are at the very opposite ends, and they are going to release something called a spindle fiber to attach to the middle of the chromosomes. I'm going to make my spindle fibers black. Now on the picture here, the spindle fibers are attaching to the middle of the chromosomes, which is called the centromere. And that gets its name because it's the center of it. And then finally, as I kind of mentioned already, the centrioles are on opposite sides of the cell. The third step of mitosis is called anaphase. Now what happens in this one, part of our three steps, is that the first thing that happens is that the chromosomes are split into two. And they splint at the centromere there. So instead of our X, now they're kind of each their own sideways V as they're being pulled apart to opposite sides. And that's the second fact that happens during anaphase, is that the chromosomes are being pulled apart to opposite sides of the cell. And they're being split apart evenly. As they're being pulled apart, they're going to start to move to opposite sides of the cell, which would be the third part of what happens during anaphase there. The spindle fibers are going to work much like a fishing pole and a fishing rod, to where the centrioles are going to be wheeling in their string for their big catch, which is the chromosomes. The final phase, a part of mitosis, is called telophase. And what the three steps that happen in this one is that we end up getting cytokinesis to happen, which is that pinching of the cytoplasm in the middle to a part where it eventually becomes two new cells at the very end. That's the first thing that happens. The second thing that happens is that we end up getting the chromosomes to go from their packaged form, which would be, our brick model that we've been using here, back into the usable DNA. It unpacks itself. It's done moving, so much like the luggage, it ends up taking out its clothes again and unpacking all the stuff from the boxes. And last but not least, we end up getting the nucleus to reappear. So like before, where we had the nucleus start to divide and split up, we get the nucleus to come back together. Alright, for this next part of the notes, I'm going to have you explain how chromatin becomes our chromosomes using a whole bunch of different terminology. Pretty much we already gone over this in the video, so I'll kind of make this quick. Originally, DNA is in the form of our chromatin. It's this unpackaged kind of mess here. When the cell divides, it wants to turn that into our packaged form, which would be our chromosomes. Now, by the time it gets divided, or to the division part, it's already made a copy of it, and that's usually in our X part. Chromosomes themselves consist of two rods called sister chromatids. So each one of these is a chromatid, and they make a chromosome. They are held together in the middle through a centromere, which I'm going to represent. So there's two bricks here. This tape is going to represent the centromere. It'll go in the middle here so that they are held together. This is so that they are not damaged during the process of moving here. And finally, once the cell division completes, this chromosome would unpack themselves back into the chromatin. Now, the final last question is kind of the big hitter, is why? Why do we care? And I'll give you 3 reasons about why we should care about this whole mitosis process. The first thing is that without it, we wouldn't be able to grow. We would stay the same size that we were when we were born, so we'd be very itty-bitty. But then we'd grow up as we get more cells. So the more cells we get, the taller that we are. The second thing is that they repair injuries whenever you're cut. The scab forms because new cells end up forming over the place of it. And the final thing is that our cells die. Depending on the type of cell, they have different ranges of their lifespans. Some cells, like blood cells, die daily. Other cells die weekly. Other cells, don't die, like your brain cells. Typically, you don't want them to die because it takes a long time for brain cells to get refreshed and made new ones up. So it all depends on the type of cell, but cells do die, and when they die, we need those to replace. Because if they're not replaced, then we die as humans.